Move on to your Bordetella species. So as you can see, this is the gram stain. So the members of this genus are small gram-negative bacilli or coccobacilli, and all are obligate aerobic bacteria, and they grow best at 35 degrees to 37 degrees. Bordetella species do not ferment carbohydrates, they oxidize amino acids, and are relatively inactive in biochemical test systems. They produce catalase, although this is variable in your Bordetella pertussis. The other Bordetella species, except for, again, Bordetella pertussis, are less fastidious and grow on mac agar, or media containing blood. So you always have to note that Bordetella pertussis is different from your other species in this genera. So we have your Bordetella parapertussis. This is non-toxigenic. You have your Bordetella bronchoseptica. This is the only motile species under this genera. And your Bordetella avium. Bordetella pertussis, also known as your Bordet gangubacillus. So it is fastidious and requires special collection, special transport system, and culture media. It is inhibited by your fatty acids, metal ions, sulfides, and peroxides, and constituents found in many media. So the media for the isolation of Bordetella pertussis requires protective substances such as your charcoal, blood, and or starch. The species possess multiple virulence factors that play a major role in the organism's pathogenicity. So here are your virulence factors. First, you have your adhesions, your autotransporters. We have your Fimbriae types 2 and 3. So this is your serotype-specific agglutinins for colonization of respiratory mucosa. Next, we have your filamentous hemagglutinin. So it mediates your adhesion to the ciliated upper respiratory tract. Again, to your ciliated upper respiratory tract. Then we have your pertactin. So it is a 69 kilodalton outer membrane protein. It mediates your eukaryotic cell binding and is highly immunogenic. So the role of both FHA and PRN is for your adhesion. So it contains the sequence of amino acids that promotes binding to sulfated glycoprotein integrins on the membranes of ciliated respiratory cells. So this also binds to CR3, and that is a glycoprotein receptor on the surface of your macrophages. The interaction leads to phagocytosis of the bacteria without initiating an oxidative burst. So that is important in the intracellular survival and replication of the bacteria. Next, we have your tracheal colonizing factor. Another one is your Bordetella resistance to killing A or BRKA. So this is a 73 kilodalton protein. Inhibits the bactericidal activity of the complement. What are the toxins for your Bordetella pertussis? So your pertussis toxin is encoded by the PTX gene, an A to B toxin related cholera toxin. So it is consisted of a toxic subunit S1 and 5 binding subunits, your S2 to S5. So 2 S4 subunits are present in each toxin molecule. Okay, and the S2 subunit binds to lactosyl ceramide. So this is the glycolipid present on your ciliated respiratory cells. Next, we have your S3 subunit. So this binds to the receptors on your phagocytic cells. Okay. This facilitates the attachment mediated by pertactin and filamentous hemagglutinin and subsequent bacterial phagocytosis. On the other hand, your S1 portion has adenosine diphosphate or ADP. So it is responsible for the ribosylating activity for the membrane surface G proteins. So these proteins regulate your adenylate cyclase activity. Next toxin that we will be talking about is your adenylate cyclase toxin. It is a bifunctional toxin that is activated in the target mammalian cell by your intracellular calmodulin. So it catalyzes the conversion of your endogenous ATP to CAMP in your eukaryotic cells. So same as your pertussis toxin. So your adenylate cyclase toxin, it inhibits your leukocyte chemotaxis, phagocytosis, killing and hemolyzes your red blood cells. So we have your dermonecrotic toxin. So it is heat labile. At high doses, this toxin causes fatal reactions in mice. We also have your tracheal cytotoxin. This is a low molecular weight cell wall peptidoglycan that has a specific affinity for ciliated epithelial cells. So at low concentrations, it causes your ciliostasis. At high concentration, it causes extrusion of ciliated cells. So the process of your tracheal cytotoxin is that it disrupts the normal clearance mechanisms in the respiratory tree and leads to the characteristic cough associated with pertussis. So the toxin also stimulates the release of the cytokine interleukin 1, which leads to fever. The lipopolysaccharide of your Bordetella pertussis produces two distinct types, one with lipid A and the other with lipid X. So both lipopolysaccharide molecules can activate the alternate complement pathway and stimulate your cytokine release. Now we will be talking about pertussis. This is also known as your whooping cough or your 100-day cough. This infection occurs after exposure to the organism through the respiratory tract and a 1 to 3 week incubation period. So it is usually 7 days, 7 to 10 days. In the catarrhal phase of your pertussis, the initial symptoms are insidious and non-specific. So you have your like sneezing, mild cough, runny nose, and sometimes conjunctivitis. In infants, they can develop apnea or respiratory distress or both. In this stage, it is highly communicable because the vast amount of the organism in the respiratory tract. The next phase, we have your paroxysmal phase. So the defining characteristic of this phase is that there is sudden onset of severe repetitive coughing followed by the distinct feature, which is a whoop at the end of the coughing spell. So unlike the catarrhal phase, it's non-specific. It's very general, like sneezing and mild cough. In here, there is this distinct characterization, which is your repetitive coughing with a whoop at the end of the coughing spell. So the whooping sound is caused by the rapid gasp of air following the prolonged coughing. Last, we have your convalescent phase. This generally starts on its fourth week from the beginning of the disease. So coughing is significantly decreased in severity and frequency. Then it will take several weeks to several months for your complete recovery from the disease. So for the lab diagnosis, this is what we use. Your Burdett Genko agar, your Regan low charcoal medium, and your direct fluorescence antibodies. This is a summary or a table for your test and medium for the different species under your Bordetella genera. Now we have our Spirilum minor or your Spirilum major. So as you can see in this photograph, it is very distinct in your gram stain. Your Spirilum minor and Spirilum major is a gram-negative helical bacteria. It is aerobic. And what you have to remember here is that it causes your spirillary rat bite fever or Sudoku. So its sign and symptoms is similar to your S. moniliformis, but swollen lymph nodes are prominent. So it is more predictable febrile episodes. Yeah,